Hi, my name is Gwen and I am a second year CMB PhD student. In this video, I'll be presenting an infographic of my project, which is to evaluate the interactions between natural killer cells and immunosuppressive cells in the hypoxic tumor microenvironment. Natural killer cells, or NK cells, are innate immune cells that play a role in eliminating tumor. And here's a diagram that highlights the various sites where NK cells can be found in our body. This includes blood, where NK cells circulate, the bone marrow, where they develop, and also the tumor, where they take action. Note that the oxygen levels are in these various sites are different, with the tumor having the lowest oxygen condition, or also known as a hypoxic condition. NK cell-based immunotherapies are extensively studied in clinics, and they show promising potentials in treating hematologic malignancies like leukemia. Two examples shown here are isolated donors-derived NK cells and also induced pluripotent stem cells-derived NK cells that are used to treat patients in clinical trials. Although there has been encouraging progress made, NK cell therapies in treating solid tumors are still limited. And this is mainly due to the challenges faced by NK cells within the tumor microenvironment, as pictured here, that is complex, is hypoxic, and is also highly immunosuppressive, meaning there are tumor-promoting cell types that live within the tumor microenvironment and they act against NK cells. A better understanding of the hypoxia-dependent mechanisms involved in the regulation of NK cell functions will be critical to improve NK cell immunotherapies that target solid tumors. Therefore, the goal of my research is to investigate how hypoxia alters the inhibitory potential of immunosuppressive cells, specifically the myeloid-derived suppressor cells, or MDSCs, on NK cell tumor cytotoxicity. The avatar is a four-chambered incubator system that can model physiologic oxygen and pressure conditions in the blood, the bone marrow, and also in tumors. By using this advanced incubator system, in vitro experiments performed can mimic in vivo settings. And for my research specifically, the co-culture of MDSCs and NK cells in the hypoxic chamber will create a setting that resembles the tumor microenvironment, which will then allow me to evaluate how NK cell functions are regulated within that environment. Flow cytometry and also IncuCyte live cell imaging system are the approaches used to examine NK cell proliferation and cytotoxic suppression by MDSCs. On the left here is an example of a flow analysis that shows fluorescently labeled NK cells assessed by dye dilution, which corresponds to proliferation. So basically, these peaks here are showing each generation of proliferating NK cells. And here on the right is a picture of the IncuCyte imaging device that performs real-time quantitative live cells imaging. Tumor cells are fluorescently labeled and cultured with NK cells with or without MDSCs, and the reduction in the number of fluorescently labeled tumor cells indicate that NK cells are killing the tumor. And therefore, the degree of reduction in tumor cells will then tell us if NK cells can kill better or worse with or without MDSCs. I have obtained some preliminary data which is not in this infographic, but basically it showed that higher suppression of NK cell proliferation and cytotoxic abilities by MDSCs in the hypoxic condition as compared to normal oxygen conditions. So by diving deeper into the mechanism of how these interactions occur, I can then attempt to block the crosstalk between these cells to prevent suppression. Ultimately, I hope my research can answer some questions that will help improve NK cell therapies that specifically target solid tumors. Thank you for watching my video. If you have any questions or comments, feel free to reach out to me through my email address listed below.